Morning, everyone. I'd like to welcome you to the second installment of the Royal Tyrrell Museum Speaker Series, which is sponsored by Royal Tyrrell Museum Cooperating Society. The work of the Royal Tyrrell Museum Paleontology takes place throughout the provinces known as Alberta. We gratefully recognize and honor Indigenous peoples' long-standing connections to these lands. Today's speaker is Dr. Stephanie Drumheller. Dr. Drumheller is an assistant teaching professor in the Department of Earth, Environment, and Planetary Sciences at the University of Tennessee. She received her PhD from the University of Iowa in 2013, and her research brings together ichnology, which is the study of fossilized behavioral traces, and taphonomy, which is the study of the processes that move, modify, or destroy organic remains after death, to ask questions about diet, feeding strategies, and preservation in the reptile fossil record. And today, Dr. Pierce is going to enlighten us on how to make a dinosaur mummy. Sorry. It's Dr. Drumheller.
Thank you. All right. As you just heard, I would consider myself to be a taphonomist. So the very quick and dirty definition of taphonomy is everything that happens to an organism between when it dies and when we find it. And if that sounds a little bit like, the plot of half of the police procedurals out on TV right now, it's true. The field of taphonomy is sort of a big umbrella term that has paleontology within it, but also forensic anthropology, zooarchaeology. A lot of us are sort of working on similar things, even though my timeline is a lot longer than people working on the forensic side. But if you think about it, there are a lot of different things that contribute to which animals end up in the fossil record and which ones don't. Are we talking about a dinosaur that was extremely rapidly buried, so it was all still nicely put together? Are we talking about something that lay around on the landscape and things scavenged it and the wind and the rain beat down, and eventually you just get one sort of sad little bone that's left behind all by itself? I usually like those sad little bones off by themselves because those are the ones that often have an awful lot of modification that tells us the story of that path. How did you become a fossil in the first place? So when I first got started in all of this, I was looking at bite marks, which is something that has been studied extensively on sort of the anthropology side of things. Bite marks are obviously direct evidence of things like feeding behavior and fighting, competition in the fossil record. And so they can tell us an awful lot about behaviors, but there's been a huge amount of research done on mammals and very little research done on anybody else, which makes sense because, again, a lot of this was being done in anthropology. Some of the oldest bite mark research can go all the way back to William Buckland in 1823, and I love these old stories because we could never get away with writing them, but he was basically like, I kept finding these bones in a cave, and they looked like somebody chewed on them, so I went to a traveling menagerie, and I threw some bones in and let the hyenas chew on them, and they absolutely look the same, done. Which I joke, and I make fun of the paper because it does really read like that. But it's kind of still what we do. We call that actualistic research. You are going out and observing things that are happening in the present day to try to figure out any patterns that you can, and then using that as a prediction to go back into the fossil record. So I was going in and wanting to look at crocodilians, because we love using crocodilians as proxies for a variety of extinct reptilian groups, but they're also really fantastic in their own right. And it just so happened that about the same time that I started working on this on the paleontological side of things, my colleague Jackson Zhao started working on them on the sort of anthro side of things. So we were all sort of coming together and looking at this all around the same time. So this is little baby graduate student Stephanie collecting some of my samples at the St. Augustine Alligator Farm in Florida. It's not a farm. I didn't name it. I don't know. It's a zoo. But they're famous for having almost all of the living species all together in one place, and they love researchers. They love letting us come in and work with their animals. So I was taking in things like partially butchered cow legs and partially butchered pig legs, and I would throw them in the enclosure. And then we would take them back again, which is, of course, the more exciting part of that equation, because they did not want to give us back the steak that we had just given them. So it was Florida, obviously, so they had a huge collection of American alligators and then smaller collections from the other species. So we did a really big collection at the individual level. which is really convenient. It's something that you can't necessarily do in the wild because I can go back to their veterinary records and say, this animal is this old, this long, and weighs this much. Perfect. And then see if there's any connection to any of those variables. But then we also did group feeding, which was going to be a little bit more natural behavior. We just throw the cow legs in the enclosure and let them do whatever they want for an extended period of time. This is also why I tend to put the staff of the St. Augustine Alligator Farm sort of front and center in my acknowledgments whenever I talk about these studies, because I was definitely in the enclosure with the animals, and they're just like, yeah, no, you go stand in the corner with your camera, and we're going to go maybe get bitten by the alligators. Like, fortunately, nobody did, but I love my lab mates from grad school. They would take bets while I was gone on how many fingers and toes I would come back with. It's like, that was not a guaranteed metric. Once I got them back, you got to clean up the bones, get all the soft tissue off to see what the marks look like underneath. And fortunately, we were able to find some nice diagnostic marks that let us know if we're looking at something like a crocodilian bite mark. They have very conical teeth, but with this prominent ridge called a carina on it, and it leaves a little subscore in the mark, and lots of details that we don't need to worry about today that means, yes, we can look at our modern crocs and we can go back and find similar things in the fossil record. So the next thing on deck was to go do that. And this name is going to come up again here in a little bit. Clint Boyd and I were both grad students at the time, and he was the first person to sort of reach out to me and say, I think I have some croc bites. You want to come look at them? And the hilarious thing about this one is it felt like cheating because it actually had an embedded tooth. This is a little juvenile Fescalosaur. And so I basically show up, I look at the fossils, and I point at the tooth, and I'm like, that major bite mark. Give me my co-authorship. It was a little bit more complicated than that. We actually CT scanned it to figure out what the shape was. But also, on the right side, you can see we did have some of those nice characteristic bite marks, even in the absence of the embedded tooth. So awesome. Compare that to my modern sample. Here we go. And a fair amount of my research has been continuing with this, trying to get a bigger sample size of things like crocodiliform, so crocs and all of their closest fossil relatives, all of their bite marks. And then we try to match that up with things like snout shape, because we have this great campfire story that we tell about crocodilians, where the ones that have the very sort of blunt, rounded snouts, they're eating hard stuff, and the ones with the big, heavy, triangular snouts are eating bigger prey. And does that actually line up with what we're seeing in the fossil record? And especially when we start throwing some of the real weird of the fossil record at it, because the fastest way to make me twitch is to say something along the lines of like, oh yes, crocodiles, unchanged since the time of the dinosaurs. I'm like, have you looked at some of these fossils? The ones today are sort of conserved, but that's because they're the last survivors after these patterns of ice ages. You look at the croc fossil record, and there's all kinds of strange stuff floating around that we don't have anymore. But even so, even though, I guess you could say my heart initially started within the crocs, I started getting a little bit further and further away from my wheelhouse. So then a colleague of mine reached out and said, well, I have some bite marks, but they're not crocodiliforms. They're distant croc cousins from the Triassic. Would you like to look at those? I'm like, yeah, okay. So what you're seeing right here are actually phytosaur bite marks on a Rauisuchian. So the Rauisuchian is the thing up top. The phytosaur itself, if you fell back through time and mistook it for a croc, you could be forgiven. They have a very similar shape because they were probably doing something very similar in their ecosystem, but they're only distantly related. And then some of my colleagues reached out and said, well, what about dinosaur bite marks? You want to work on dinosaur bite marks? I'm like, those are archosaurs still. I can play with dinosaurs. Let's do this. So I was going out and looking at some of the Morrison Formation specimens and looking at bite marks that they have on their bones. And then colleagues started showing up with fish bite marks. And I am so far out in the weeds, I'm not sure I can find my way home again. This is, I'm not a fish paleontologist. Both of these I'm going to have to update the slides soon because they're both sort of in the publication pipeline. So spoilers, I suppose, but those papers will hopefully be out soon. So when Clint Boyd, remember Clint Boyd, baby's first publication back in grad school? When he called me up and said, I have another bite mark project for you. I was like, cool, okay, what is it? And then he got a little cagey on me, and he's like, well, I don't really want to tell you much about the project until you agree to work on it. And I'm like, Clint, you're not going to get me to help you hide a body, are you? And he's like, no. It's a dinosaur mummy. I'm like, what? Okay, yeah, sure. So I was invited up to Bismarck, North Dakota to look at Dakota. Dakota is an Edmontosaurus. It's one of those situations where it's probably a Nectans, given the age and the location, but we don't have the head. We have everything else, we don't have the head. And this was not my normal thing that I work on. Usually I'm looking at those really hideous, beat-up bones that have been stomped on and chewed on and exposed to wind and rain, and this was the prettiest fossil I had ever seen in my life at that point. I feel like it took me about 24 hours to just emotionally come to terms with this thing, because it's just gorgeous. But from a taphonomic point of view, it's weird. So let's talk a little bit about soft tissues in the fossil record for a second. The majority of times when you hear paleontologists talk about soft tissue preservation, it's in the context of what we call Lagerstätten, which is a fancy term that just means fossil mother lode, unusually well-preserved fossil deposit, and that means the whole deposit. So usually what you're seeing is there's some kind of weird chemistry going on in that environment that is suppressing microbial activity and keeping out things like larger predators and scavengers, so that decomposition really, really slows down, and you will eventually get that thing buried and then start to fossilize. So think really absurdly low oxygen, or really ridiculously salty, or in some extreme cases, like freezing. So think about all of those mammoths that keep coming out of the permafrost, or perhaps extremely rapid drying. Dinosaur mummies have always been a problem with this whole understanding of how that kind of preservation works, because they're often not found in what we would consider a Lagerstätten-style deposit. Most of the fossils that are found in the same area will just be bones. They'll maybe be disarticulated and, my usual sort of beat-up, sad fossils. But then every once in a while, you'll get sometimes one or sometimes a small cluster of these animals that seem to have the soft tissues on them. We've known about them for a good long while, but the number of them out there in the world is actually pretty small, so they're considered to be fairly rare. The majority of the research that has previously gone into these has been describing the soft tissues. And this is not me casting shade on that, because it's incredibly interesting and very important. We're trying to understand what these things looked like, how that might have affected how they lived. But I'm a taphonomist. I want to know how on earth that thing fossilized in the first place. And that side of the literature has been a little, we'll say, less well explored until fairly recently. So you get these proposed mechanisms that to a taphonomist read like magic. Magic happened. And I'm like, no, we're going to have to unpack this. So you'll get this explanation that they had to be rapidly buried. And a lot of times people will say, well, maybe it was a flood. Maybe this thing died on a riverbank and then was just immediately swept away in a flood and buried. So that's going to prevent things like predators and scavengers coming and chewing on them, and weathering and UV radiation and all of that fun stuff, and microbes from breaking them down. But then you'll also see they all look desiccated. They all look dried out. So that means they had to lay around on the surface for a very long time until they dried out. So they were instantaneously buried while somehow also being exposed and drying out, and just, what? But again, that was not the gist of those papers, so they could just say, oh, something weird happened. Let's talk about the skin. But I wanted to talk about that. Because those things seem to be mutually exclusive, right? Like, you can't be super rapidly buried, but also exposed and dried. Which gets us to our initial specimen. It's nicknamed Dakota. Again, it's an Edmontosaurus. It has a little bit of a confusing history in terms of the repository where it is, so you might actually see some previous papers under MRF3, which is the Marmouth Research Foundation. It was then transferred to the North Dakota Geological Survey in 2008, but the ink didn't dry on the actual paperwork until 2017, so nobody was really working on it in that intervening time. It is from the Hell Creek Formation, so about 67 million years ago. And right up here, you should start to see our first little wrinkle to that explanation of somehow we had maybe incredibly rapid drying. It was a super arid environment. This is fluvial, so river sediments through deltaic deposits. Okay, so coastal area, forested coastal plain. Hell Creek is most famous for its dinosaurs. However, it's just absolutely chock full of the entire ecosystem. So you get terrestrial, freshwater, brackish environments all mixing together. Great big dinosaurs all the way down to tiny little invertebrates and things like that. So it's a very diverse ecosystem, and it would have been pretty wet. So there's our sort of we have an issue here. Now, Dakota had not been fully prepped when it was transferred to the North Dakota Geological Survey. So I always have to give them a shout out here. Mindy Householder is the one in the picture, but also Becky Barnes were the two preparators who were working on Dakota. And I feel like preparators do not get the love they deserve because like they're the real heroes here. This thing was in a giant, really hard block of rock and we couldn't see anything. And they're the ones who go in and take all of that nonsense off the fossil so we can see what's underneath. And they were the ones that came back to the researchers and said, there's something weird going on with the skin. Like there's holes in it and gouges. You should get somebody to look at that. Now, I don't even know how many hours we're up to at this point, just person hours working on Dakota, but the punchline is it's not done yet. So what you're seeing right here is, at least as of 2022, yellow areas are parts of Dakota that have been fully prepared, so we can see all the skin nicely. Red areas are parts of the body that we know there's skin there, but it hasn't been fully prepped, so it's sort of peeking out around the matrix. Black areas are missing, so we don't have the head, alas. We're missing one arm and a couple bits of the tail. And then the area that isn't shaded in at all, there's no skin apparent there. There's just exposed bone. And we absolutely got mocked when the paper came out. So, did you mean to make the Belgian flag? I'm like, no, but it's good colors. Good job, Belgium, I guess. But I was brought in to look at these marks because usually when I get randomly called in to go somewhere, it's because somebody bit somebody else. It's starting to be now because there's some dinosaur skin, but this is why that second part has come into play. This is actually Dakota's hand. So you've got tip of the hand over here, that nice mitten that we all know and love for members of these groups. This hand, it was a natural break. I didn't snap it off, I promise. But it would perfectly fit if you just held your tongue right and got it at a funky angle in the stage of the micro-CT scanner at the University of Texas. So we were able to take a peek on the inside. And that's where things started to get a little interesting for us. The first thing is the bones on the inside of the hand are not deformed. Right? They haven't been flattened in any way. But you can also see a little bit along the top margin of that scan, the three-dimensionality of the skin. So it's not just that traditional way that we often see mummies described where this animal died and it fell into some nice, squishy mud and then left an impression of the skin and then that decomposed away and then something else came in and replaced it. It seems like we might actually be looking at some type of 3D preservation. Minerals came in and replaced those soft tissues. So that was intriguing. And we were able to actually go through and look at those bones and how they relate to the overlying tissues. So we're having a lot of fun with that scan. They tried to scan the bigger body block in one of the big industrial scanners, and it wasn't giving us terribly great resolution. But this one was a lot of fun. Now again, there was a fair amount of research done on this animal before it was transferred to North Dakota, and certainly before I became part of the project. But this was all stuff that had been reported previously. There's potentially things like biomarkers that have been recorded from it. We're still sort of looking into that. We're also looking at chemical analysis, and I'll touch on the super preliminary results for that at the very end of my talk. But this wasn't what I was called in for. I was called in for the weird holes. So okay, let's talk about those for a second. This is the little fifth digit, little pinky finger on the hand, and there was a little arc of these circles in the skin right there. Moving over onto the tail, which is again beautifully preserved, you have what looks like almost a tear in the skin up here, and then these large sort of gouges, these raking marks down the side of the skin. Now, they were asking me to identify who did this, who actually bit this animal. And that's where I started to get a little bit, we'll say angsty, a little worried, because I was familiar with the forensics literature. If you do any kind of taphonomic research, there's so much overlap, you end up going and reading papers from other disciplines. So I knew that despite what some of the TV shows would have you believe, bite mark evidence in soft tissue hasn't really been admissible in a court context for a very long time. And it makes a lot of sense if you think about it. And this is where I have to say, I'm going to get a little bit gross. I'm going to try to just keep it a little bit gross, so apologies in advance. But if something bites something else and that animal survives, then you have healing, and the healing can then warp what that bite mark looks like. But if you have something bite another animal and then that animal dies, decomposition will then warp the bite mark. So trying to match up bite marks on soft tissue with whoever did it in the first place is really, really challenging. And there's been some research that says maybe we can figure out major clade differentiation, but even that is a little in the forensic context. So I was starting to get a little worried. And then finally I got to breathe a bit of a sigh of relief, because bone is far more rigid. When you have an impression of an impacting tooth left behind on bone, it tends to actually preserve the shape of that. And lo and behold, we actually did get some fun bite marks on the bone. I'm going to zoom into a couple of these, but I'll have to give a little bit of an explanation. So this is the humerus, so this part of the arm sticking up out of that skin. Please do not Google degloving injury. Let me explain it to you instead, and then just be satisfied with that. So if you think about like a long set of, opera gloves, if you started to take them off and turn them inside out, that's a degloving injury. This right here is actually the inside surface of the skin that has all kinds of holes and tears along the margins, as if somebody bit onto it and peeled it open to get at the nice muscle underneath. Because very few animals are really terribly interested in skin as a food resource. They want what's under it. It's more nutritious, more bang for your buck. And lo and behold, we do have some fun bite marks on them. They're just highlighted right here, adding some color to it. These are called hook scores for probably obvious reasons. We line that up with what's called an inertial feeding strategy. Crocs and also theropod dinosaurs and a lot of groups that eat like this are messy in terms of eating. They don't have very good table manners, but they will grab onto stuff and then maybe shake their heads. If you're a crocodilian, you might do that famous death roll. But what that means is the teeth will sort of start moving in one direction and then take off and go in a different direction. So that's not diagnostic of Crocs, but we did have a bisected pit. That's that little blue one. That's that nice ridge on the tooth that we see in our Crocs. Okay, cool. At least the arm, whoever bit the arm was a Croc. So I could tell them that, and they could be happy with me for a little bit. But the other trick was I kept sneaking off and looking at the other parts of the body that they had. And it was sort of funny, because then like Clint and Mindy would sort of chase me back over to the bite marks. They're like, there's no bite marks on this. Come over here. But these two areas in particular, I kept getting drawn back to. This one's a little maybe hard to figure out what you're seeing. This is a foot, so you have the toes coming out here. But then this rounded part at the bottom, essentially what you're seeing there is when this animal was alive, it would have a big fleshy pad underneath its foot, kind of like an elephant to help distribute that weight, because they are awfully big. And what's happening is the skin is now sort of pulled out to the side, and it's completely empty. It looked like a deflated balloon. And then over here, what you're seeing is the base of the tail, which was also just completely deflated. We already saw those scans of the hand. We weren't seeing any internal tissues. It really was just the skeleton with the skin on top. So no muscles, no internal organs, just bones and skin. And I realized I had seen stuff like this before, this very deflated appearance. I just hadn't seen it in a dinosaur. I'd seen it in people. I'm not going to be showing you those pictures, so don't worry about that. But the University of Tennessee in these circles is actually quite famous as being the first place in the world where human taphonomic research was performed in a medical legal context. Now, don't get very excited. William Bass, the founder, has retired. That's not his number. Please don't call it. But the facility is still out there and running. They are the largest facility of its kind, but more of these facilities have opened up in other parts of the world because, as you might imagine, the climate of East Tennessee is going to be very different than other parts of the world, so we would like to sample across all of them. But they have over 1,000 individuals that are already in the collections. They have over 4,000 what they call future donors, people who have said, I want my body to be donated when I pass away. And there's this whole wealth of literature done on the forensic side of things, including, did I somehow kill it again? That seems like a thing I would do. There we go. All right. Rough stages of decomposition. Now, for actual forensic anthropologists, this has been updated quite a bit, but it relies very heavily on specific weather conditions that we just don't have access to in deep time. So this first sort of quick and dirty pass at what are the predictable stages of decomposition, these are quite useful for us in paleontology. But what you'll see is even just looking at this model, which was done with pigs, we already have some predictions in place that were starting to play out with what we were seeing in the mummies. First of all, during active decomposition, this is when all of the insects and the small scavengers get really excited about your set of remains. They will take advantage of natural body openings, so things like the mouth, things like the nose. But even early on in the literature, they were saying, or injuries. So they don't really care, they just want to get to that yummier stuff on the inside. But then even the dry stage of decomposition, which can take months, years to get to and then persist, they mention dry out skin and other dermal tissues, so things like nails, things like hair, my reptile friends, things like scales. So we started looking at this and saying, it sounds like scavenging might actually sort of unintuitively promote mummification. And there were some studies out there, case studies in the forensics literature suggesting that. So what we were trying to say with Dakota is that the deal with partial scavenging, if you have injuries that compromise the skin, and again, we're going to get a little gross for a second, but this allows sort of an exit route for all of the gases and liquids related to decomposition to get out of the body, along with those microbes that are so destructive. So you're basically emptying out the skin envelope and leaving that behind where it can become dried out. And that can last a very long time up on the surface. So everything we were citing was from the forensic literature, and we knew this. So we needed a little bit more to sort of convince our colleagues in paleontology. So we were also looking at the sediments surrounding the mummy, because the initial interpretation was, again, maybe this thing got swept up in a flood, and that's how it got rapidly buried and done. So we actually went back to the sediment, and instead of getting bigger class, which if you think about a flood event, higher energy water can move bigger things, right? You have a raging river, it's going to pick up a bigger rock and carry it along. If you have very slow-moving, gentle water, it's only going to pick up little stuff. So if you had a catastrophic flood event, you would expect the size of your grains to maybe pick up, or at least get some bigger ones in the mix. It actually gets smaller the closer you get. So it really does seem like whatever happened here, it lay around on the surface for a while, being sort of slowly, gently covered. So our explanation was, we do not know if our croc friends, and it might have actually been theropods in the tail, there were no bony bite marks on the tail, alas. However this animal died, it was at least partially scavenged by at least 2 actors. And then our little micro-scavenger friends, our little invertebrates came in and they joined the party, the microbes were in there, and eventually you end up with this deflated skin envelope over the skeleton that gets slowly buried. And we got a couple testy reviews on that paper, if we're just being very honest with one another. But one of the main arguments was, why are you only citing mammal studies? And I'm like, because there aren't any reptile studies. Okay, fine. So, this is the usual way science works, is, wow, there's a big old hole in our data set. Maybe we should fill it. The simple fact of it is that a huge amount of work has been done on mammals, and we, over on the reptile side of things, will sometimes take that research and then maybe sort of uncritically blast it onto other groups. So this was a foundational study by Berensmeyer in 1978 looking at bone weathering. What happens to a bone if you just leave it out on the landscape and you let UV and wind and rain do its job? And we all cite it. And I'm trying to, I'm not calling out other researchers because I'm hanging out in the et al. Of this paper right here. We all then say, oh yes, our specimen here is a Berensmeyer weathering stage 2 or whatever. Is that justified? Can we actually take this mammalian work and just apply it to reptiles? So you do what you do. You go grab yourself a grad student and you throw them at the problem. So this is Hannah Maddox. She actually just recently defended her thesis with us, and she's moved on to the South Dakota School of Mines. But that was her whole project. So Hannah, bless her heart, did 2 runs from Tennessee all the way down to Florida and back to pick up 60 Argentinian black and white tegus. Now these animals were not specifically killed for us. These are very popular in the pet trade, and they're also a very problematic invasive species in the Florida Everglades. So there's a lot of different federal agencies that are basically trying to get that population under control. They are carnivorous, and they'll just eat everything, including protected groups. It also means that when researchers call up and say, like, can you just give us an enormous pile of lizards? They're like, yes, please, just take them away. So we brought them back up. We put them in these boxes, and I also remember that Buckland paper where people used to get away with writing really goofy methods sections, and now we have to at least sound professional. It is a 2x4 box with chicken wire on the outside to keep the coyotes out of it, and you have to make that sound really well thought through. But it's also highly necessary. So what you're seeing right here is a very grumpy vulture, because we did have these larger predators and scavengers coming back who would like to carry off our animals. We would love to maybe see that later on, but if they actually physically ran off with all of Hannah's tegus, there goes her thesis. So chicken wire to let the things like the insects in, but to keep the larger ones out. Sorry, Mr. Valter. They're still out there. They're decomposing away. I'm actually doing this over on our agricultural campus associated with the University of Tennessee. But these are just showing some of the time lapse of what happens. So you start off, they get placed. We have 30 that were our warm weather run that were set out sort of mid-May and then left to go through a Tennessee summer. We have a cold weather run that was sent out in February instead and let to be snowed on a little bit before everything went along. And we have been watching what happens. Just because we do have a lot of people who work in forensics at UT, I reached out to a few of them and said, I'm doing this, would you like to come play ball? Because you hear forensics and you immediately think of the human angle, but there's also things like wildlife forensics, and they're having the same problem that we are, where there's a lot of mammal studies, but if you're dealing with like a poaching case or a smuggling case involving reptiles, so if you look up here, this was a very, very recent survey of all the different groups that have been sampled for what we call the necrobiome, the microbes that are present on an organism after death and then proceeding through decomposition. So if you look down the names over here, they're probably kind of small for those of you in the back, but we have a bunch of humans, kangaroo, bunch of pigs. Pigs are a very popular proxy for human decomposition. Beaver, rabbit, trout, mouse. It's mammals, a whole bunch of mammals and one fish. So we actually got another graduate student whose name is also Hannah. So there's a lot of Hannahs on this project. Her name is Hannah Noel. She's working over in the Ag campus looking at soil science and chemistry. So she was coming out and she was taking swabs of the tegus. And she was also taking soil samples and then clippings from the plants. Because when things decompose, you can think of it as nutrients are now getting put out in the surrounding sediment. So it goes into the sediment, but then it also gets picked up in the leaves of the plants. And can you tell things like a clandestine grave based off of just plant clippings? So it's interesting because a lot of the chemical change that they were seeing in the soil, this lines up quite nicely with what you would predict from pretty much the mammal data set. So that's cool. But the necrobiome, we did have a couple interesting things that popped up. The first one is our friendly reminder that sometimes I and all of my colleagues in paleontology forget, is we have this idea of burial being some sort of magic cure-all. Like if we can just stick it underground, then no more decomposition, it's protected. But the majority of the microbes that actually kick this process off are not external things coming in, they're your gut flora. They're the stuff that's in your digestive tract already, and basically it's like, well, if you're not gonna feed me, then I'm wrecking the place. So all of the colors that you see pointed out here, those are those things that we call them endogenous. Those are the endogenous bacteria, the gut microbes taking over. But then on the fungal side of things, especially in the dry stage of decomposition, we saw some fun stuff pick up that seems to be specialized groups that are breaking down keratin in much higher spikes than what you see in mammals. And we certainly have keratin in our bodies. It's what your fingernails are made of and your hair. But reptile scales, you have a lot more. So it makes sense that maybe these guys are coming in and having a little bit of a party there at the end, but it also helps explain why that skin kept lasting so much longer. And then this is very exciting. It's very gross, but I'm having a great time. Let's start adding more taxa to it. So this last summer, we actually put out 4 American alligators and 2 African dwarf crocodiles to sort of join in the fun. So we're still going out and looking at our tegus, but now we're looking at these guys as well. The idea here was they're a lot bigger, and they also have osteoderms, those bones inside the skin, the armor that we see in some of these groups, and how does that start to affect some of these things? There are a lot of things that we're seeing that we feel a lot comfier about that mammal literature now, but there's a couple things that got kind of weird, sort of unexpected. The first one is that long-term retention of so much skin. And again, apologies, a little gross. You come to a taphonomy talk, it's going to be a little bit gross. This is the same animal. It was one of our smaller tegus. And you can see it, so we have day 7, day 25, day 92. Look at the position of the tail and look at the position of the head. What was going on is all of those internal tissues were gone, so you just have the skin, and it basically adhered to the underlying bone. And then it would dry up, and that skin would start to scrunch. And then it would rain, or there would be like a really heavy dew in the morning, and it would get wet and it would relax. And then it would dry again, and it would scrunch. So we start to see the head thrown back, and we start to see the tail thrown back, and that's starting to be that very classic, what we sometimes call the death posture, that you have some really famous examples of in your own gallery outside. Head thrown back, tail thrown back. There's a lot of debate about that in the paleo literature. You'll see everything from fluvial activity all the way to the literal agonized death throes of the animal, or the reason why they would be posed like that. And at least with our friends here, we saw it in the tegus. We haven't seen it in the crocs because they went a little bit too fast. The fun thing with our croc sample, though, is how long those things like the osteoderms got held together, but then also how much variation that we had. So what you're seeing here is one of the little African dwarf crocs, and these guys were, 4-ish feet when they were alive. That's our biggest alligator. He was our big boy. Oh, sorry. Metric. Little over a meter on this one, a little over 2 meters on this one. It's the, what do I tell my colleagues? And then, wait, you're not in the States, Stephanie. Get it right. So that one over there is our big boy. He was a little over 2 meters. And you'll notice this is the same time. This is the same day. They were set out on the same day. He's still covered in skin. A lot of it. Over here, it's a little hard to see, but there's a little bit of skin left on the hands and the feet and the end of the tail, and that's kind of it. So what's going on? The alligators were donated to us by a nuisance alligator hunter because, yes, that's a thing in the American Southeast. And the crocodiles actually came from a zoo that donated them to us. We didn't actually realize until it showed up that the crocs were going to be necropsied first, which makes sense in retrospect. They want to make sure that there was nothing funky going on with this animal before they turn it over to the mad scientist to throw in her field. But it means that they were cut open and a lot of the stuff on the inside had been removed, kind of like a scavenger might have done. So if you're taking out all those guts that have all those microbes in it and you're throwing them away, what does it do to decomposition? That's probably the direction that we're trying to go next, is following, sort of chasing that one down the line. But a lot of the variation that we're seeing is along the lines of what does that long-term retention of skin do? We get postural shifts. We get some interesting patterns of disarticulation because as long as everything's inside the skin, it's held nicely together. But then in the heads, we get a lot of strange patterns because sometimes the skin will adhere to the bone and then you have a nice skull that's all put together. Lizards don't have very fused up skulls. So what happens when it doesn't fuse to it is essentially that's the first thing to disarticulate. And then you start wondering about things like, why can't we ever find sauropod heads? Might be something like this. So it starts to explain some of the odd patterns that we've noticed in the fossil record. But to loop back to our friend Dakota for a second, we have these assumptions that we've been making, things like it has to have been buried rapidly. Rapidly is a dangerous word in geology because when we're talking about paleontology, we're usually thinking in deep time. Hundreds, thousands, tens of thousands, millions of years. So like when I'm sitting in a red light and it's taking over 60 seconds, I'm like, this is the longest red light ever. And then when I'm actually doing my job as a paleontologist, I'm like, 10,000 years, that's like a rounding error, who cares? It's a really weird way to have to think about time because the Earth is really ridiculously old. So what do we even mean by rapid burial? So we keep saying the word rapid and then thinking in our day-to-day lives rapid, like flood, insta-buried. A lot of evidence for slow sedimentation and extensive drying, and looks like that skin can actually last. We're into year 3 on the first batch of tegus, and all of them have at least a little bit of skin left on them, which was not what we were expecting going in. Desiccation has to be really, really fast, or otherwise you have to protect these things from scavenging, because if anybody comes and eats your carcass, no more mummy. Except we have direct evidence of something coming along and munching on Dakota. And if we look at the forensics literature and this preliminary work done by Hannah and case studies in forensics, that might actually help with the mummification side of things. And unusual preservation means that dinosaur mummies are exceptionally rare, but they're actually not. Now, this is sort of the disheartening side of my presentation. The fun thing about coming to places like the Royal Tyrrell is you come in and you're like, I would like to see your mummies that I know about. And usually what ends up happening is then the curator is like, well, here are these, and here's 30 more that are in collections that have never been described. So, and it's always sort of a fun, exciting, like, what are we going to find when we show up? A couple of things that we've been finding is that we have a horrible, sneaking suspicion that skin is actually pretty common out there, and we've been accidentally destroying it. Because from a preparation point of view, and again, I'll let Mindy, really get on her soapbox about this, but this is not me trying to be critical. It's just we're trained to go all the way down to the bone. And if you don't have a reason to suspect that there's skin there, You're not going to keep an eye out for it. And if we're assuming it's incredibly rare, why would you even keep an eye out for it? Just expose the bones. We keep going to museums, and there'll be like a little patch of skin, and then there'll be this very suspicious dark halo around the rest of the body, and we're like, well, we'll sample what we still have. The other thing we're finding is a lot of the previous literature that's been attempting to explain mummy fossilization, we keep looking for a silver bullet. We keep looking for one explanation that can apply to all of them, even though we already know that's a little bit. Mummy and mummification in this kind of a context is a very sort of loose, casual term that is applied to stuff like Dakota, stuff like the specimens that are out in your own gallery. But I've also seen things like those frozen mammoths being called mummies before, so things that were exposed to extreme cold or extreme dryness, cave deposit stuff. This is not the same pathway to preservation. There's different things happening, and then we're just calling them all a mummy. So there was a paper that came out by Postural and colleagues looking at some of the Wyoming specimens and then comparing that out there. We're looking at that, and that's our example that we use to sort of illustrate that is something like Leonardo, this Brachylophosaurus, where we really are looking at a situation where the skin is not preserved there in 3 dimensions. You had a very complex series of events to get that nice impression. They call it death masking, and it's involving clay minerals and the microenvironment related to microbes. It's a very cool process, and it seems to work for things like Leonardo. We also have stuff like Dakota. You also get things like this specimen that seems to have been an equivalent of what we sometimes would think of as a bog body. You get deposited in anoxic conditions. Anoxics are very low oxygen. They basically got pickled, and that's how they fossilized. And then you get things like Dakota, where there does seem to be some three-dimensionality to the preservation, but we're calling it deflation and desiccation. The skin is there, the bones are there, none of the other internal tissues are there, and it's all just sort of this collapsed, empty skin envelope with a skeleton underneath. And that seems to be pretty common. But we're out here trying to now figure out how these things fossilize. So I've been very focused on the death to burial side of things. This idea that skin can actually persist on the landscape for a surprisingly long amount of time. This idea that scavenging might unintuitively help this mummification process along. And it also explains why, now that we're starting to look, there's a lot of skin out there. It might not be the whole body, but in terms of patches of skin, we find them on dinosaurs quite a bit. Clint Boyd, then, the other half of our duo on this project, is really focusing a lot more on this burial to fossilization. So some of my research trip up here this week has been scouting for him so he can bring his little handheld XRF up. Handheld XRF is a really quick and dirty way to figure out what elements are present. If we want really detailed data, it's destructive. We have to take a little piece and grind it into powder and stick it in the machine. And as a general rule, people don't like it when you show up to their very beautiful fossil and ask to smash it to bits. So we like to do this instead. It's a good way to sort of target, and it's a quick and dirty pattern. And then if we need to do something more destructive, we can then say, like, these areas are interesting. Are there any natural breaks or things that have fallen off the specimen that might be hidden in a drawer somewhere that we can run away with? This is Dakota. That's actually the little handheld XRF. Everywhere that you see a little green dot on the jacket over here, that's somewhere that he was taking a measurement. But after testing poor Dakota half to death, we've then been going out to a bunch of other specimens to build a big data set to try to figure out if we can tease out some chemical patterns, like what is happening to these guys. So there's Leonardo. There's Hannah hanging out with me again, working on that one. This was us going to Museum of the Rockies. That's not the XRF, that's a 3D surface scanner, so we were getting a 3D model to then match up with where we were taking the samples. And then so much math. This is still in progress. I keep fussing at Clint because I'm like, you have to stop eventually. You have to actually analyze a data set. You can't just keep making it bigger and bigger. And he's like, don't talk to me. I have to make it even bigger than this. So that's what he's coming up here to do. But the idea is we're trying to figure out what is the chemistry of the surrounding rock, what's going on with the skin, what's going on with the bone, and are they all the same, or are there predictable differences? And if there are some patterns, like do we see ones in this formation look like this, and ones in this formation look like that, and is that because of the matrix, or is there something else going on? It's complicated. It's, that's, science, I suppose. But for Dakota itself, we're not just doing the XRF, we actually are doing sort of the full-on, full-court press, trying to do all of the, samples of destructive analysis, things like SEM, EDS, to really look at what minerals are present and what might be going on here. And it is interesting because it does seem like in at least some parts of this animal, we might actually have some interesting 3D replacement style preservations. Like the skin is not there anymore, but these minerals have come in and taken the skin's place and maintained some of that structure. So there's so much research left to do, both on sort of the front end with my decomposition research, but also on the back end where we're starting to see several different research teams that are getting really excited about looking at the geochemistry of these fossils to really understand how they happened, partially to understand the ones we have, but then also to potentially find even more, because every time we find one of these mummies, it's just this complete treasure trove of new information about those animals. And with that, thank you all for coming and being hopefully only a little bit grossed out. I tried not to jump scare you guys with anything too bad. Thanks for coming, and if you have any questions, I would be happy to answer them.
Okay, well, thanks, Stephanie. Okay, so we have time for questions.
Hi. So I'm not entirely sure how to phrase this. So my question is more like, do we know how often scavengers will just completely disarticulate a skeleton or like take it away or a body or something? Like how, I guess I was surprised that there are these bodies that are out for a while, even the ones that are partially scavenged, and they're still largely in a big piece or most of these pieces.
That is, like, I think every paleontologist at some point in our careers has this like dark night of the soul where we have to think about all of the information that's been lost. Because if you think about just the rock cycle and the water cycle and plate tectonics, like all of these things that make our planet so dynamic, what's going on is everything is trying to recycle everything else all of the time. So everything about this is trying to break down that organism, get those nutrients that were in it, disperse it back out into the environment, and then your food web keeps going. So the fossil record is basically that process. For some reason, there was a hiccup. We didn't quite manage to completely break this thing down and get the nutrients back out there in the wild. It did get buried. Some sort of chemical reaction started happening that stabilized it. I've seen, it's one of these sort of off-the-cuff estimates, but it's less than 1% of all things that ever lived on this planet actually ended up fossilized. And we haven't found them all yet. I think the current metric is just dinosaurs. We're not talking about the entire rest of the diversity of all living things on Earth, just dinosaurs. About one new species every single week is being published right now. And it seems to actually be picking up steam. They joke and call my generation the Jurassic Park generation because like I was tweens when that came out. I'm telling on myself age-wise. But you got this real explosion of interest along with that. And there are more paleontologists working in the world today than there have been at any other point in the history of the science. So yeah, we're still finding stuff, but there's still a lot of stuff to find. The fact of it is that they are rare because all fossils, to one extent or another, are kind of rare. Our concern is they're not as rare as we previously thought. So just if you're looking at some of these animals and you're in the right environment, keep an eye out. But the overwhelming majority of remains that are out on a landscape are absolutely going to be almost completely broken down by predators and scavengers and then exposed long enough that the wind and the rain and everything else finishes the job. And it's the whole dust to dust thing. You're eventually going to completely break down all those nutrients and they're gone. And that is just something that we have to sit with and be very uncomfy about when we are doing our reconstructions of these ecosystems. But when you get that weird chemistry that lets fossilization happen, sometimes you get those nice lenses, so you are getting nice sampling within it. That's my unhelpful but hopefully very honest answer to your question, I suppose. Did it answer your question, or do you have a follow-up?
I'm more, I guess, wondering if there have been any, I guess, animal forensic studies on like how many things are getting taken apart and how many are just like staying still or staying in mostly one piece.
Yes. So it's highly variable based on the environment and the groups you're looking at. We call them live dead studies, and it's exactly what it sounds like. So you go out, and one of the most famous ones is, again, Berensmeyer's research in Amboseli National Park. You go out there and you get your little clipboard and you say, okay, who do I see living and walking around out on the landscape? Okay, who do I see that's still present? If I'm just looking at bodies, I'm just looking at bones. And then if you're feeling extra fancy, you can actually do things like trenching and transects and say, okay, what survived till burial? Really happy data sets where our live dead studies make us feel kind of comfy. like 50%, right? You can get all the way down to, and then it's just gone. Like, you want to study fossilization of things that lived up in the highlands, like on the peak of a mountain? I am very sorry. If you want to study things like the tropics, it's a biodiversity hotspot in the present day. It's also really, really warm, really, really wet, and the microbes love that nonsense. Fossil record's not great. You get, some things, but even just terrestrial to marine. Most of my colleagues back at Tennessee who are in paleontology work on marine fossils, and they're always sort of turning their nose up. It's like, oh yeah, dinosaurs, terrestrial fossil record, that's garbage. And I'm like, shush. Like, you're not wrong, but also stop it. You're hurting all three of my feelings. But it's true. The trick there, though, is, again, that's actualistic research. We have to go look at all these different environments, and we have to look at all these different groups, and the numbers are all different depending on what you're interested in. So that's where things like hard tissue bias comes into play. If you have a skeleton, you have a hard mineralized shell, you're far more likely to end up in the fossil record than an earthworm. So we are in taphonomy. That's a lot of the research that's gone into it historically. With taphonomy, there was a shift in about the 1980s where we were trying to get positive information out of it. What can this tell us about the environment? And prior to that, it's all just, it felt like raining on everybody's parade. I say that like I was alive before the 1980s, but you look at the literature and it's very much like, here's what's lost and nobody can do anything, and it's very woe is us. But the shift in trying to figure out that positive side of things is then saying, okay, if we have the right system and the right organisms that we think we should have about 50% preservation, that actually, if you sample it, gives you a pretty good idea of what the life assemblage was, so we're good. And if you go to your fossil assemblage and it's maybe not a great system for that, there's still work to be done. You can still describe those fossils, figure out where they fit in the tree of life, but maybe then extrapolating out a big paleoecological signal is not what you should be doing with this data set. You got to think about how the data set might be messing with your underlying science, and that's true in every science, not just paleo. It's complicated. It's unhappily complicated, unfortunately.
I'm a technical lead on the Kasky hadrosaur, which is a big mummy, hadrosaur mummy in Dinosaur Provincial Park.
Yes.
And we are working on that in the field, and you've alluded to technicians, and thank you for the shout out for the technicians being underappreciated.
Very much so,
Yeah.
and none of us would be having this conversation
But the
without
commonality with technicians in the field and in the lab is glue. We use all kinds of glue and solvents and things, and I'd like to know your thoughts on how that, which we are squirting gleefully into the rock to stabilize cracks and crumbly bone and crumbly skin, how that is affecting your geochemical studies moving forward. And I think we really could use some guidance moving forward. If we find another mummy in the future, maybe we should be saving samples or not gluing certain parts. I think we really could use some guidance.
I'm going to slip Mindy's contact information to you. No, I'm going to call her this afternoon and say, like, hey, Mindy, that glue paper that we keep telling you to finish? So this is something that we've noticed. And certainly when you're looking at specimens that have been found and prepared more recently, generally technicians are really good about keeping a record of this is the glue I used and these are the areas I used it on and here's our record. But when you're looking at something like that Osborne mummy from the 19-teens, Lord only knows it was probably a boiled down horse. Like, there's no record and it's really dubious. So what we have been finding is that glue is I think the way Clint has described it is it's like a glaze on top of the fossil. And with things like handheld XRF, you're really just getting surface data. So it is putting a little bit of a barrier in between. And some of the elements, the signal is strong enough that it makes it through, and some are not. Now, the really disconcerting part is that you have the chemical formula of the glue itself, but then we also end up getting things like there's a big chlorine spike that happens, and that's because it's basically leaching chlorine out of the plastic containers it's been stored in, and then putting that. So you don't necessarily immediately think it should be there because there's no chlorine in the chemical formula of this glue. So what Mindy has been doing, and that I'm going to have to go needle her about, is once we started realizing that this was a problem, she got some of, I think it's actually the matrix samples, like leftover chunks of the matrix from Dakota, I think is what she's working with, and essentially went through and took the XRF and went zap, 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 zap. and then went through and put glue on it all, and then zap, 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 and then put another coat, because, sometimes one isn't good enough. And I think she went up to like 3 or 4, and then, just for extra spice, then she's like, okay, acetone. Because it's reversible, right? It's like it never happened. And then zap, zap, zap all the way down the line again. And what we're seeing is there are predictable chemical shifts. So if you know the glue is there, you can at least say, okay, ignore the chlorine. Like whatever chlorine is doing, you just ignore it. It doesn't exist. It's contaminated. But these things are probably going to come out, and you can still look at those and talk about the pattern. These things are masked. So there are patterns that we can tease out to figure it out, but in their heart of hearts, what Clint really wants is a nice, clean, pristine sample. So it's the suggestion that they've been making, and I've been to some mini talks on the subject because obviously this is her wheelhouse. I'm just the one who came afterwards. But if you are able to retain some parts and say, no glue has ever touched this, that'll make the geochemist very happy. Of course, the standard, if you do use glue, just tell us where and tell us what, and so we can assess that. A lot of times, the chemical testing, the glue will only soak in so far, so if you have something that wasn't on a surface that was coated, you can at least get away from that and maybe sample there. But anytime glues are introduced, it is changing the signal. And we're trying to get a handle on what that change is so we can account for it. Because, glue is very necessary for, I'm not saying don't ever use glue. That's clearly not in the cards. But anytime that we can sort of figure out, let's preserve this little bit, and then we can use this part later, is terribly useful for this kind of research. Then you get into the historic stuff, and it's like, this shellac is made of bugs? I got to learn all kinds of fun new things during her project. Like, we're putting liquefied bugs on our stuff. That was all 100 years ago. Nobody does that anymore. But she can still get online. She's been trying to find modern sources for some of these historic glues just to say what on earth to put in her sample and test. So it's interesting. I think there was sort of a wince that went across the room when she presented at the methods section at one of the more recent Society of Vertebrate Paleontology, because it is a problem, but it's a known problem, and it's one that if we're aware of it, we can start to get a handle on it, but it does change the underlying data, is my very wordy answer, sorry. So, no worries. But no, absolutely, if you're interested in working on a specific specimen, reach out to Mindy. She's fantastic, and she's happy to help, and she's talked with other researchers who are working on skin, so she's happy to share everything that she's figured.
Yeah, I was wondering, is there any good example of Ceratopsian or sauropod or theropod mummies, or just mostly hadrosaurs?
They are out there. There's a Ceratopsian, like the Houston Museum. Most of the skin was prepared away, but they saved sections of it that are on display next to it. There are some examples of Diplodocus out there that have skin. There are some disheartening examples where there's field photos of Tyrannosaurid fossils with skin in place that are now prepped off. There's an awful lot of mummy research that you just kind of want to go scream into your pillow afterwards. There's 1 and possibly 2 mummies at the bottom of the North Atlantic because of World War I. There's just all kinds of data sets where you're like, well, we used to have that mummy, and now it's gone. That has been a certain area of discussion, why do we keep finding them on hadrosaurs? Why is it just a hadrosaur skin party and everybody else seems to be rare? And it seems to be a combination of factors. It is possible that they did have slightly thicker, more durable skin. It is also possible, because we love calling them things like the cows of the Mesozoic, there's just so many of them out there that even if it's a very small percentage that fossilize this way, you're more likely to find more of them because there's just tons of them. There's also, unfortunately, we're not looking for them, and we might be accidentally destroying them. So, okay, remember my first love, my crocs that I keep joking that I'm trying to sneak back to, and they keep dragging me back into dinosaurs? There's a hand stuck on Dakota's hip. And this was all before it made it to North Dakota. But there's a very conspicuous black silhouette around that hand, and they went straight through it to get to the dinosaur underneath. And I'm like, that was a croc mummy. So I think they're out there. We're just not realizing what we have and maybe accidentally destroying it. So I would say for anyone who's doing any kind of field work, if you are digging anything up and there is a weird color or texture change as you get closer to the bones, it's worth it to just slow down and just take an extra look, because maybe that's what you're seeing. But there's also sort of a grade of preservation as well. So it might be that this was skin, but you can't really see anything, and it's just sort of a sad stain in that layer, and you're not going to get morphology or anything out of it. But just take a peek, just in case. I suspect we're going to start finding a lot more, which is really cool to think about, but then also kind of disheartening about the stuff that has accidentally been lost.
This is sort of picking up on what you were just talking about. Have you, talking about like whether hadrosaur skin is special or if it's just because there's a lot of them, have you done any of that or are thinking about doing any of that with your taphonomic studies with the animals that you're leaving out to rot? Like, does different animals have different skin compositions that react differently?
Remember my grad student, Hannah, who's off at South Dakota. She only did a master's with me. That was 2-year project. Her PhD, what she's working on now is looking at things exposed on the surface, but also burying them, and then looking at the geochemistry of what happens to the skin, where it starts and where it goes once you start having those early stages of diagenetic alteration. So there's been, again, some of that research has been done, but it's very mammal-centric, again. So she's wanting to get samples across a bunch of different groups. We're trying to get more samples. Like I would love to get more crocs. I would love to throw out some birds. Maybe get some of the weird morphologies, like some snakes and turtles. I would just like to have everything, honestly, but I'm pretty certain the ag campus would get very angry at me if I just took over, a couple acres of nothing but this. But the work is ongoing, and there are other labs who are also doing the kind of stuff that Clint's doing the deep dives like what we're doing on Dakota, but looking at other specimens that were fossilized in other ways. So like we're working on it, but this is definitely sort of a recent trend. Because again, a lot of the historic literature on the mummies it wasn't the question being asked. But we're trying. We're working on it. It just takes a little bit of time, especially for these long-term decomposition studies to see what happens. So.
Okay, thank you,
Thank you.
Stephanie.